Good day, everyone. Now we are going to proceed with our next lesson entitled Non-Fermentative Gram-Negative Bacteria, Haemophilus species, and Miscellaneous Gram-Negative Bacteria. Alright, class, let's warm up by talking about non-fermentative gram-negative bacilli, or your NFOs. These organisms are the opposite of Enterobacteriaceae in many ways. So first, they cannot ferment carbohydrates. If you run a TSI, it will stay K over K, meaning alkaline over alkaline, and it has no sugar fermentation whatsoever. In the OF test, they fail to acidify the closed tube. Why? Because they prefer oxygen. These organisms are strict aerobes. They do poorly in anaerobic conditions. Most NFOs oxidize glucose in the presence of oxygen, meaning they produce acids only aerobically. Some are even asaccharolithic, which means no sugar metabolism at all. A major key point and something you should always think of when differentiating from Enterobacterales, NFOs are usually oxidase positive, and of course they love moist environments. Think humidifiers, sinks, catheters. Almost all are naturally resistant to several antibiotics, which makes them clinically important even though many are environmental organisms. Now, aside from the basic biochemical profile, NFOs differ from other gram-negative bacilli in three more ways. First is the motility. Some have polar flagella while others don't. Next is the pigmentation. Many NFOs produce distinct pigments that will help to identify them. And lastly, growth on your McConkey agar. Some will grow on McConkey and others won't. So whenever you encounter a gram-negative bacilli that is oxidase positive, non-fermentative, and has some interesting pigment or motility, start thinking, this might be an NFO. For NFOs, we use a few consistent tests across organisms. McConkey agar or your MAC, does it grow? If yes, does it ferment lactose? Usually non-lactose fermenters. Oxidase test, most NFOs are oxidase positive. OF glucose test, this is crucial for confirming oxidative versus fermentative metabolism. These three tests are your starting point in any NFO workup. So let's zoom in on the OF test. It's designed specifically for NFOs. It uses 0.2% peptone, meaning it minimizes protein metabolism and highlights sugar oxidation. The indicator used is bromthymol blue, which changes color in the presence of acid. So here's the trick. Oxidizers will only produce acid in the open tube. Fermenters will show acid in both tubes. This test is indispensable for differentiating NFOs from your Enterobacterales. When we think about NFOs routinely isolated in the hospital, four major groups dominate. We have Pseudomonas aeruginosa, Acinetobacter species, Burkholderia species, and your Stenotrophomonas maltophilia. Pseudomonas aeruginosa alone accounts for 90% of all clinically significant NFO isolates. So as future medical laboratory scientists professionals, this is the organism you'll encounter the most. Let's jump into Pseudomonas. It is the king of your NFOs. So the characteristics include gram-negative bacilli or cocobacilli, strictly aerobic, motile with polar flagellum, except Burkholderia malay, oxidase positive, except your P. luteolus and your P. oryzae habitans. They are catalase positive. They grow on McConkey as a non-lactose fermenter, and then these traits make Pseudomonas easy to differentiate from Enterobacterales. So this is where Pseudomonas gets scary for hospitals because they love pools, hot tubs, catheters, humidifiers, and the sinks. So they are found in plants, soil, water, basically everywhere moist. Though not normal flora, they cause 5 to 15% of all nosocomial infections, especially respiratory tract infections. They also grow well at 37 to 42 degrees Celsius. So feverish patients, no problem. They thrive there. Now let's talk about how they damage the host. Pseudomonas has many potent virulence factors such as your endotoxin or your lipopolysaccharide, motility and pili, capsule, phospholipases, type 3 secretion system, a variety of exotoxins including proteases, hemolysins, lecithinase, elastase, DNase, but the star of the show is exotoxin A because it blocks protein synthesis similar to diphtheria toxin. That alone makes Pseudomonas a formidable pathogen. In patients with cystic fibrosis, Pseudomonas aeruginosa often produces mucoid colonies. Why? Because they overproduce alginate. Alginate is a thick polysaccharide that forms a slime layer. So this slime enhances adhesion, immune invasion, biofilm development, and once this organism colonizes cystic fibrosis lungs, it becomes extremely difficult to eradicate. So if you plate Pseudomonas aeruginosa on a blood agar, you will see large, flat, spreading colonies, often beta hemolytic, a distinctive metallic sheen, and a characteristic grape-like, fruity, or corn tortilla-like odor. That smell is due to 2-aminoacetophenone, a signature compound of this organism. You will never forget that smell after your first encounter. Trust me. Now let's talk about one of the most iconic features of Pseudomonas aeruginosa, its green coloration. This distinctive color comes from two pigments working together. Pyocyanin, which imparts blue color, pyoverdine, which imparts yellow to green or yellow-green color. When combined, they create that characteristic blue-green or green sheen on agar. Clinically, this pigment mixture is so characteristic that you can often suspect Pseudomonas aeruginosa just by color before any biochemical test. Let's dig deeper into these pigments. So the first one is pyocyanin. It is water soluble, it is produced by most strains, it's extractable in chloroform, it has antibiotic-like activity, which means inhibiting other bacteria. It also contributes to oxidative stress in host tissues. The second pigment is pyoverdine. So it is also water soluble. It is also known as your fluorescein. It gives colonies a yellow-green or a yellow-brown appearance, which functions as a siderophore. Siderophore will help the organism acquire iron. Together, they make Pseudomonas aeruginosa visually unmistakable. Now for the core biochemical profile, these help us confirm identification. For TSI, it is K over K, which means non-fermenter of any sugars. Arginine dehydrolase positive, it grows at 42 degrees Celsius. They are citrate positive, and they are also positive for acetamide utilization. And a very important selective medium is your citrimide agar. This medium inhibits most other bacteria while enhancing pigment production. So if you see growth plus the green pigment plus the fruity odor, you're almost certainly looking at Pseudomonas aeruginosa. So this organism is a classic opportunistic pathogen. When tissues are compromised, Pseudomonas aeruginosa jumps in. Key diseases include wound and burn infections, which is famous for producing the blue pus, nosocomial infections, especially ventilator-associated pneumonia. We also have hot tub folliculitis, or the so-called jacuzzi or the hot tub syndrome. Pseudomonas can also cause nail infections, especially with artificial nails. Otitis externa, known as the swimmer's itch. S- uh, swimmer's ear, rather. Swimmer's itch is for Mycobacterium marinum. Please don't be confused with those terms. And then we also have eczema gangrenosum. It is a necrotic skin lesion which is seen in immunocompromised patients. So anywhere there is moisture plus the weakened immune defenses, so think of Pseudomonas. Now, here's the challenge. Pseudomonas aeruginosa is innately resistant to many antimicrobials, so treatment options are limited. However, they are often susceptible to aminoglycosides, semi-synthetic penicillins like your piperacillin, third and fourth generation cephalosporins such as your ceftazidime, cefepime, and then combination therapy is common to prevent resistance. And as future medtech professionals, you will appreciate how crucial antimicrobial susceptibility testing or AST is for this organism. So this table transitions us to another group of important NFOs, the Borcholderia species. So they share some similarities with Pseudomonas but also have unique features and pathogenic roles. So this section includes Burkholderia cepacia, Burkholderia malay, Burkholderia pseudomallei, and others. So let's go through them one by one. So the first up is Burkholderia cepacia, which is clinically important, especially in cystic fibrosis patients. The key characteristics are the following. So it grows on your sheep's blood agar but may lose viability within 3 to 4 days if not subcultured. They can also grow on your McConkey agar. They require selective media to suppress Pseudomonas and improve recovery, such as PC agar or the Pseudomonas cepacia agar. We have the OFBL or the OF base plus the polymyxin B plus the bacitracin plus your lactose. And then we also have your BCSA. BCSA, it is your Burkholderia cepacia selective agar, so which is the best for isolation. When they grow on your agar, colonies are non-wrinkled, which helps distinguish them from Pseudomonas tutzeri. A few more fun facts regarding Burkholderia cepacia. They do not fluoresce, and intrinsically, they are resistant to aminoglycosides, polymyxins. Many strains also resist beta-lactams. So again, AST is absolutely necessary. This organism is a major concern in cystic fibrosis patients, where it can cause rapid lung deterioration. Now let's look at Burkholderia malay, the causative agent of glanders. It is a rare but serious infection that primarily affects horses. Why are we talking about it in medical technology? Because it is considered a potential bioterrorism agent. It is non-motile, it is resistant to polymyxin, so while uncommon, it's important from a public health and biodefense standpoint. So this one is clinically much more relevant. So we have Burkholderia pseudomallei, the agent of melioidosis. If you encounter a non-fermenter that forms wrinkled colonies and shows bipolar staining like a safety pin, suspect Burkholderia pseudomallei. On ashdown medium with colistin, colonies turn the pink thanks to neutral red. It also has a very characteristic earthy odor. But remember, do not sniff plates in the lab. like ever. We only mention this because older texts refer to it. Next in the Burkholderia group is your Burkholderia gladioli or gladioli. This organism primarily known as a plant pathogen, but it can infect humans, especially immunocompromised patients. So the key facts of Burkholderia gladioli, it resembles Burkholderia cepacia complex. So they can also be seen in cystic fibrosis and chronic granulomatous disease or CGD patients. They can be recovered from blood and tissue in severe infections. The colony characteristics of these organisms are the following. So it produces a yellow pigment after 48 to 72 hours. So they are motile with one or two polar flagella. They are catalase positive. They are urease positive. They oxidize glucose, so they are not asaccharyolytic. They grow on McConkey agar. They are mannitol positive. They are decarboxylase negative. They are oxidase negative. So some strains may appear weakly positive. So this is one of those non-fermenting organisms that can surprise you by appearing harmless but causing disease in the right host. So here's an interesting laboratory challenge. Some Pseudomonas and Burkholderia species tend to resist decolorization during gram staining. So the result, they may retain the crystal violet, making them appear gram positive or gram variable. This can easily lead to misidentification, especially for newer laboratory personnel. So when you see an organism that looks gram positive but behaves like an unfermenter, reconsider your first impression. But this table right here shows you other organisms aside from Pseudomonas and Burkholderia species. So we have your Acinetobacter baumannii, you have your Acinetobacter iwufi, you have your Acinetobacter hemolyticus. So as you can see, we already have here their specific host, their disease association, and as well as their biochemical characteristics. So kindly familiarize this table, everyone. Next, we also have your Alcaligenes fecalis. We also have your Acinetobacter calcoaceticus, and as well as your Moraxella lacunata. Moraxella lacunata produces lacunae or pitting of the agar. But please also take note, aside from Moraxella lacunata, your Echinella corridens will also pit the agar. Okay, so as shown in the picture on the right. Now it's time to look at the third most common NFO in clinical laboratories. We have your Stenotropomonas maltophilia. This organism was originally seen as a harmless saprophyte or colonizer, but now we know it can cause serious infections. It is not considered normal flora, but it can rapidly colonize the respiratory tract, especially in hospitalized patients receiving antimicrobials such as your cephalosporins, penicillins, carbapenems, aminoglycosides. Why those? Because Stenotropomonas maltophilia is naturally resistant to many of them, meaning those antibiotics wipe out competitors, and this organism fills the ecological niche. So this organism can cause a variety of infections, but the most commonly noted ones include endocarditis, especially in IV drug users or patients with heart surgery. We also have wound infections, including cellulitis and ecthyma gangrenosum-like lesions. And then we also have bacteremia, and rarely meningitis and urinary tract infection. Interestingly, while usually not associated with respiratory tract infections, it can be isolated from cystic fibrosis patients. Though is less common than with your Pseudomonas. For identification in the laboratory, here is the classic profile of your Stenotrophomonas maltophilia. So they are oxidase negative, they are non-fermentative. Colonies may look bluish on your McConkey agar. They are catalase positive, DNase positive, esquilene hydrolysis positive, gelatin hydrolysis positive, lysine decarboxylase positive, and this is important. The drug of choice is trimethoprim sulfamethoxazole. This organism is resistant to many other antibiotics, so AST is critical. Now we shift gears to another major group, the Haemophilus species. The name Haemophilus literally means blood-loving, and that's because these organisms require certain factors found in our blood, specifically the X factor and the V factor. X factor is also known as your hemin or the hematin, while the V factor is also known as your NAD or the nicotinamide adenine dinucleotide. So some species need both, others only need the V factor. So here's a key trick, everyone. Species whose names start with para, like Haemophilus parainfluenzae, require only the V factor. So let's break down how Haemophilus behaves on different blood agars. On horse or rabbit blood agar, some Haemophilus species show hemolysis. On sheep's blood agar, V factor is not available because sheep RBCs contain NADase. It is an enzyme that destroys the NAD. Therefore, V factor dependent species do not grow on sheep's blood agar. Para species grow only when an external source of V factor is present. But here's the practical shortcut. Chocolate agar is the preferred medium for Haemophilus. So chocolate agar or chalk is essentially a blood agar that has been heated to around 80 degrees Celsius. So heating does two key things. First is that it releases the X factor, which is the hemin or the hematin, from your red blood cells. And then the second one is that it inactivates the NADase enzyme, so protecting the V factor or the NAD from being destroyed. So modern laboratories may also use synthetic chocolate agar which contains NAD, iron, vitamin B12, thiamine, magnesium, cysteine, glucose, glutamine. So this provides optimal conditions for the fastidious Haemophilus species. Now let's focus specifically on the X factor. It is a degradation product of your hemoglobin. So this is heat stable, known chemically as hemin or hematin, so which contains iron. So sources would include bacteria such as Staphylococcus aureus, yeast, potato and yeast extract. So if an organism requires X factor, it cannot synthesize it and must extract it from the environment. So the V factor is quite different, also known as your NAD or the coenzyme 1, because they are heat labile, so they are destroyed easily by the enzyme NADase, not readily available from intact RBCs. So this is why certain media fail to support Haemophilus growth unless the RBCs are lysed or unless another organism supplies the V factor. And this sets up our next concept, which is satellitism. Okay, sorry for that. Alright class, let's talk about satellitism. One of the classic laboratory phenomena you must know for Haemophilus. So satellitism occurs in V factor requiring Haemophilus species such as Haemophilus influenzae, Haemophilus parainfluenzae. So here's how it works. Some organisms like Staphylococcus aureus, Streptococcus pneumoniae, and Neisseria species naturally release V factor or the NAD as a byproduct of their metabolism. On your SBA, V factor is not available, so Haemophilus cannot grow. unless a helper organism is present. The Haemophilus species will then grow as tiny satellite colonies so around the helper organism because it provides the V factor. So this is seen as a halo or a zone of enhanced growth surrounding, let's say for example, Staphylococcus aureus streaks. So remembering this high yield point, so all clinically significant Haemophilus species except Haemophilus ducreae require V factor and show satellitism. So if you have an isolate that grows around Staphylococcus aureus on a sheep's blood agar, you're likely dealing with Haemophilus influenzae or another V factor dependent species. Haemophilus ducreae is the exception because it needs only X factor and not V factor. So Haemophilus species actually make up about 10% of the normal microbiota of healthy adults' upper respiratory tract. Most common normal flora species, we have your Haemophilus parainfluenzae. We also have the non-encapsulated Haemophilus influenzae or the NTHI to a lesser extent. So the colonization of your Haemophilus influenzae begins in infancy. So this is important because colonization can transition into infection, especially in children or patients with respiratory diseases. In diagnosing Haemophilus infections, specimen collection is absolutely critical because these bacteria are extremely fastidious and die quickly outside the host. So common specimens include blood, CSF, middle ear fluid, respiratory specimens, conjunctival swabs, genital specimens. For Haemophilus ducreae, lower respiratory infections often require bronchial washings for optimal recovery. Please remember this one, everyone. Specimens must be processed immediately because Haemophilus is highly susceptible to drying. Conventional media alone aren't enough because they lack the X and V factors. So we rely on chocolate agar, which is the most commonly used culture media. It is incubated at 33 to 37 degrees Celsius, which requires 5 to 10% of carbon dioxide. The growth is visible in 18 to 24 hours. The second one is your chocolate agar plus bacitracin, which is 300 milligrams per deciliter in concentration. This is excellent for isolating Haemophilus influenzae because bacitracin suppresses normal respiratory flora. So a big clue for Haemophilus identification is gram-negative pleomorphic cocobacilli on your chalk with no growth on SBA or MAC. So this is a classic differential pattern. So this part outlines the general workflow or the algorithm for identifying Haemophilus species in the laboratory. So we have primary culture, plate on your sheep's blood agar, chocolate, and your McConkey. And then examine the colony morphology. On your chocolate agar, they will appear tannish, moist, smooth, convex colonies. On your sheep's blood agar, growth only if satellitism is present because V factor is present. And then McConkey, no growth. For the microscopy, they appear gram-negative pleomorphic cocobacilli, encapsulated strains, show clear halos. So for the workup tests or the biochemical tests, you can use oxidase test, X and V factor dependency test, the D-ALA or the porphyrin test, the traditional biochemical test, and as well as hemolysis patterns on your sheep's blood agar. So this algorithm ensures accurate differentiation of species such as Haemophilus influenzae, Haemophilus parainfluenzae, and Haemophilus ducreae. So this is one of the most fundamental tests for identifying Haemophilus. So your X and V factor test. So you place discs impregnated with the X factor or the hemin, then the V factor or the NAD. And then we have an XV combination disc. Then you're going to observe which discs support growth. So interpretation, let's say for example, growth only around the XV, we have your Haemophilus influenzae. If the growth is only around V and XV, that's your Haemophilus parainfluenzae. And then if there is growth only around the X, so therefore it is Haemophilus ducreae. So this pattern is practically a fingerprint for species identification. Next, we have your D-ALA or the porphyrin test. So this test determines whether an organism can produce X factor on its own. So the mechanism or the principle, the organism is given delta aminolevulinic acid, and then if it can convert ALA into porphyrins, it means it does not require the X factor. So are we going to interpret it positive, presence of fluorescence? Organisms can synthesize hemine, so meaning it does not need X factor. Usually, those Haemophilus with para on this species. So negative, there's no fluorescence. Organisms need X factor. So think of Haemophilus influenzae or Haemophilus ducreae. So this is a key confirmatory test. So next, we also have your X and V factor test, which is somewhat similar to those what we have discussed earlier. If you have Haemophilus influenzae, then there is growth on your XV impregnated disc. Haemophilus aegypticus, there is growth on your XV impregnated discs. And then for Haemophilus parainfluenzae, growth on both V and XV impregnated disc. And then for Hemolyticus, growth only on XV. And then for parahemolyticus, growth on V and XV. So this picture shows the classic school of fish appearance on gram stain for Haemophilus ducreae, the one on the upper right corner. So cells appear arranged side by side. So in parallel rows, so like small fish swimming in the same direction. Okay, so the colonies are located the cells are located at the bottom part of this presentation. So this distinctive morphology helps identify Haemophilus ducreae in genital ulcer disease, or the so-called GUD cases. Now let's talk about Haemophilus ducreae more. It can cause chancroid. Chancroid is also known as the soft chancre, which is a painful genital ulcer, as you can see in the picture. So compare this with syphilis, which causes a hard, non-painful chancre. So here's an important clinical reminder, everyone. So chancroid increases the risk of transmitting other sexually transmitted diseases, especially HIV, syphilis, herpes virus. So patients presenting with genital ulcers must be tested for multiple STDs. Now class, let's shift to a very important group of fastidious gram-negative bacilli, which is known collectively as the HACEK group. So HACEK is an acronym representing H for Haemophilus species, A for Aggregatibacter, C for Cardiobacterium, E for Echinella, K for Kingella. So what do they have in common? They are all fastidious, which means they are slow-growing organisms that require increased carbon dioxide and are often part of the oral flora. Clinically, the biggest takeaway is this. The HACEK group has a strong predilection for infecting damaged or prosthetic heart valves. Their well-known causes of infective endocarditis, So let's take a closer look at the common characteristics shared by HACEK organisms. First is that they are fastidious, so they require enriched media, often chocolate agar. Capnophilic, enhanced growth in 5 to 10% carbon dioxide. They are dysgonic, which means poor or slow growing, except for Haemophilus, which grows faster. So they are also part of the normal oral flora, but they can enter the bloodstream following dental manipulation, trauma, or poor oral hygiene. So because they are slow growing, they may require prolonged incubation, and sometimes they are only detected once blood cultures flag positive after several days. These organisms are notorious for their role in endocarditis, especially after dental procedures, uh, in IV drug users, in patients with congenital heart disease, in those who have prosthetic valves. So they tend to cause subacute slow progressing infections, meaning symptoms can be mild and non-specific for weeks. So when blood cultures come back positive with a slow growing gram-negative rod, the HACEK group should instantly come to mind. So while this presentation may not list each organism individually, but here are classic differentiators used in the laboratory. So for the aggregator species, so they are part of former Haemophilus aprophilus or Haemophilus paraprophilus. So they are catalase positive and they may show star-shaped colonies in older cultures. Other details are presented already in this table. The next one is the Cardiobacterium hominis. So they are rosette or flower-like gram stain morphology. They are Indo-positive. Next we have your Echinella corrodens. So they have a distinctive bleach-like odor. They also have the tendency to pit or corrode your agar. And it is associated with human bite wounds, specifically those so-called fight-bite injuries. Then lastly, we have your Kingella kingae. So they are beta hemolytic. They're associated with pediatric bone or joint infections. They can resemble Neisseria morphologically. So these traits help medical professionals narrow down the organism before confirmatory testing. So this table shows you the different HACEK members and then their different biochemical reactions on certain biochemical tests. So please familiarize everyone. So this table shows you another organism. So we have your Capnocytophaga, we have your Calimactobacterium granulomatis, Chromobacterium violaceum, and your Cardiobacterium hominis. So kindly familiarize their typical characteristics and as well as their disease association. Now, let's look at Brucella, everyone, commonly known as the Banks bacillus. These are small, non-motile, slow-growing intracellular pathogens associated with zoonotic infections. So the classic transmission routes include unpasteurized dairy, direct contact with infected animals such as cattle, goats, and pigs. So mode of transmission, inhalation, and lab-acquired infections are also possible. So Brucella species can cause brucellosis, also known as undulant fever, which is characterized by intermittent fever spikes, night sweats, hepatosplenomegaly, chronic fatigue, and please take note that this organism is highly infectious in the laboratory, requiring BSL-3 precautions. So this part shows a table of key biochemical traits. So here's the classic profile for distinguishing Brucella species. They are gram-negative cocobacilli, they are urease positive, and they are urease rapid producers. And then they are oxidase positive, they are catalase positive, they are non-motile, they require extended incubation for growth, and specialized serologic testing and molecular assays are often used for definitive identification because they grow so slowly and pose biosafety risks. So this table serves as a reminder that the world of facetious gram-negative bacilli is broad. Beyond NFOs, Haemophilus, and HACEK organisms, there are many others that require enriched media. So carbon dioxide or special atmospheric conditions. So these organisms often resemble normal flora. They grow slowly and may cause serious infections in immunocompromised hosts. Because of these features, accurate identification often depends on a combination of culture, biochemical testing, molecular methods, and clinical context. So please also familiarize everyone the different characteristics and association of your Campylobacter and your Helicobacter pylori. Now, for the Bordetella genus, it's famously associated with whooping cough, which is common for Bordetella pertussis. So the key characteristics include they are small, gram-negative cocobacilli, strict aerobes, do not ferment carbohydrates. Many species require charcoal-containing media, such as your Regan low agar. They are very sensitive to drying and toxic substances. You'll often diagnose Bordetella infections using PCR, direct fluorescent antibody, culture, although it's slow and requires special transport media. Classic laboratory features for Bordetella pertussis, they are mercury drop-like colonies, which will grow on your Bordet gengo agar. And then they are non-motile, they are urease negative, and as well as they are urease positive. So this summarizes the key characteristics of the different species of your Bordetella and their biochemical reactions. So this part likely highlights miscellaneous fastidious organisms that don't fall neatly into the major categories. So this may include your Legionella, Francisella, Gardnerella, or other rare non-fermentative gram-negative bacilli isolates. So these organisms vary widely in pathogenicity, but the common theme is that they need enriched or specialized media. They are often part of the animal reservoirs, and they can cause opportunistic or zoonotic infections while less frequently encountered. So medtechs must remain aware of them because misidentification can delay the treatment. So please take note of the following. We have your Legionella pneumophila, causative agent of your Legionnaire's disease or the Pontiac fever. We have your Legionella micdadi, the causative agent of your Pittsburgh pneumonia. Your Francisella tularensis, it's the causative agent of your tularemia. And please familiarize also other typical characteristics of the organisms listed here. So next, this is the last table, everyone. So this shows you the different organisms, so other miscellaneous organisms such as your Gardnerella vaginalis. You have your Streptobacillus moniliformis, and your Pasteurella multocida. So please also familiarize their typical characteristics and as well as their disease association. And with that, everyone, I would like to conclude our discussion including non-fermentative gram-negative bacteria, Haemophilus species, and your miscellaneous gram-negative bacteria.